So shall I start?
Yeah, sure, sir. Hello, everyone. Present here virtually. I am Nalini here, and I welcome you all for today's session, which is titled as "Use of Use of Bioinformatics Related Tools for Plant Breeding Research." And presenting this session, we have with us our today's expert, Dr. Sandeep Debnath. Dr. Sandeep Debnath is an experienced assistant professor at the Department of Genetic and Plant Breeding, Pali Shiksha Bhavan, Institute of Agriculture, Vishwa Bharati University, Sriniketan, West Bengal, India, with extensive expertise in the higher education sector. He has expert in both plant breeding and genetics. Strong education professional having a PhD in a genetic from Vidhan Chandra Vishwavidyalaya. Additionally, Dr. Debnath is active in a member of publication and conference presentations, and he has already authored in a 13 research articles, 16 patents, 2 books, and 6 books chapters. His areas of studies include bioinformatics, mutant breeding, biotechnologies, and enabled plant breeding. He has supervised 2 research projects. Without wasting much time, I am welcome to Dr. Sandeep Debnath. You may start now, sir.
Okay, so thank you everyone, and wish you a very good morning today. And the topic that I'm going to deliver a lecture on this is use of bioinformatics related tools for plant breeding research. Actually, before going to this, just a minute, I just want to, yes. So before going to the lecture, with a note I want to start, that is that I think all of us will admit that actually change is the only constant in this universe. Okay, in our planet also, change is the only constant. Always something is changing, and this is the only truth. Changing, and we are also changing ourselves to make our world better. So with the and this plant breeding as a subject, plant breeding is being practiced throughout the world since time immemorial. We can't remember. Okay, that means when that art of plant breeding was started by the humankind. And why did I say that change is the only constant? Actually, people from the very first day of plant breeding of humankind, people wanted to change the architecture of the plant, the genetic makeup of the plant. And this is the only motto of plant breeding. That means I have to grow more, and that's why I have to change the present plant that I have in my hand for cultivation. I have to change it, then with the possibility that it will yield more, that plant will yield more, and it will be able to feed the whole world. Okay, not only the humankind, the whole world, all the animal world, everything, it will be able to feed. So the first target, the very first target of that practicing of plant breeding was to achieve the food security. Actually, increase in world population is not a problem today. Okay, the world population is increasing. Okay, and it is a truth. It is, and it will increase. So first target was food security, but to achieve that, at that very first stages, nutritional security, food safety, these were not the primary concern at that time. Later, people understood, okay, we have to, not only we have to achieve the food security, we have to achieve the nutritional security, and we have with the standards of food safety. Okay, then only the civilization will be able to survive. So actually, this bottleneck theory, okay, so we had a number of germplasms, a genetic reservoir we had. You can see from this slide. Okay, so from millions of years ago that we have. So we, through a kind of bottleneck, we have just shortened out that which plant is important to me, and then people has decided, okay, this plant is to be transferred to a crop. That means for large scale cultivation. Though there are several theories that we cannot make each and every plant species edible, because among the millions of plant species, among a number of numerous numbers of plant species, edible plants are around 30,000, the number of edible plants. But if you can think very, very critically, you will find that only on based on 10 to 15 crops, the whole world is able to get their food. Okay, so only out of these so many numbers of plants, actually we have to sort it down. Okay, and this work has been done, okay, due to the course of plant breeding. Okay, the evolution of crops, including loss of genetic diversity. Actually, for breeding or for making the desirable traits, or for making a desirable plant a better idiotype, we have lost the genetic diversity. It is also a truth. Okay, due to domestications, genetic bottlenecks, and we have selected the land races, and the modern plant breeding. We have also selected from that land races also, because if you get back to the history of land races in plant breeding, land races you will find very, very region specific land races. Okay, sometimes village specific land races. So after the bottleneck effect, when was the cultivation era of land races by the farmers, at that time also the number was numerous. But with the effect of after Green Revolution, just the number has been reduced down to popular cultivars. Hardly it can be counted by hands. Okay, so this is very, very few in number after Green Revolution. So one thing that for secure the foods for all for this campaign, it is proved that we have already lost the genetic wealth. So that's why, though we have achieved the food security to a level, but also now we are also at a stage that we have to think critically that now I need those genetic wealth, those alleles back. Then because we have reached to a yield plateau. So this was the yes. Just a minute. Yes. Okay. So this is in a nutshell the plant breeders do. Okay. Suppose we have found that a line which is very elite for suppose 1 or 2 traits, that line may not be a very good yielder, but we have found a desired gene in that, and our by crossing or by any traditional means, we just want to transfer that desired gene into a recipient variety. Maybe suppose a very popular variety lost a very important character, and we are just
Dr. Devnath, slides are not changed.
I have already changed that.
No slides are being changed. First slide is shown here. No slides changing.
Oh, may I request the host that can you help me in that matter? Because I am changing my slides.
You can send me your presentation, sir. I will share.
Please, please pardon.
Actually, sir, participants are asking about slide changing. You have not changing your slides.
But I am changing my slides. So shall I share it again by closing it?
No, sir. Only slide changing. Of second page.
Yes, I am at the second page. I have already changed the slide.
Slide means, but not showing, sir.
Okay, let me do it again. Is it visible now?
Yes, sir. Visible. First page, sir.
Only first page. And now?
Sir.
Yes, visible.
Visible, sir.
Is it visible now? The second slide?
Yes, sir.
Yes, sir.
Okay. So in this diagram, you can see that in case of our traditional plant breeding methods, what we do, actually we try to introduce a gene of interest, suppose in a popular variety which have lost its some important properties. Okay, so and it is very clear from this picture. Okay, and the same logic is being applied for the genetic engineering or biotechnology part also. That means just we are changing some methods, but actually we are doing the same thing. We are trying to incorporate the desired gene. Okay, previously it was by only by crossing, but here just using some modern methods or biotechnological tools, we are just incorporating the desired gene and we are making a new variety. But something is happening. I don't know when I want to change the slide. Okay, I have again changed the slide. Is it visible now?
Yes, sir. Visible.
Okay, thank you. So the present rate of yield, the yearly yield improvement for the key crops runs from 0.8 to 1.2% only. But if you can remember what happened during Green Revolution, we are not being able to achieve that kind of jump whenever we are applying our all efforts. Okay, like we are applying biotechnological tools, we are doing molecular markers, everything we have tried, but not being able to jump or get the improvement in yield uh, in comparison to the effort that we have made till date. Okay, so that's why people are scared, the scientists are scared that whether all these techniques, everything, they will be able to generate such a crop variety or to make the changes in the crop varieties that will it be able to feed the whole world. Okay, so there are only two primary ways to increase the agricultural production on farms. Okay, this is the hypothesis. Okay, that we have to make superior crop types by deploying some genetic modification that is done by the plant breeders, or we are having another option that you just change your management methods. That means you change the package of agronomic practices that you can gain something. But whatever you do, actually, ideally, people are trying to handle both these things simultaneously, and that should be the approach. That means integration. Not only the plant breeders will change the genetic makeup, agronomists also come forward, and we have to make. That means whenever a new variety is bred, all the package of practices you have to try, and also this is done while releasing a variety, but still we are unable to achieve the goal. This is the problem. So what is the need of the day? Because we have already tried everything. Okay, need of the day is that we have to speed up the process. We have to speed up this plant breeding process, this artificial evolution process, but with no or minimal mistake. This is very important. We have to speed up the process, but we shouldn't do any kind of mistake, or we can't waste many time. Okay, so this is very important. And okay, as I was talking about the time, that means time management with no or minimal mistake, without any error, we have to manage the time. So now have a look what is actually going on in the science of plant breeding. Actually, today I think it is better to call the technology of plant breeding. Okay, because science it has already been established, and we all know the science behind the plant breeding. Actually, technology is changing day by day. Not the science behind the selection is changing. It is because science is constant. But the technology is changing, and we have to adapt the technologies, and we have to try those technologies in our plant breeding fields. So if I look at the crossbreeding process, if you see here, that means I am taking two different parents, which is a kind of elite variety, which is actually disease susceptible, and very popularly we use the backcross breeding method and you can see the percentage also. That means after 5 to 7 repeated backcrossing, you can achieve your goal. You can make your variety. It was elite in the sense of agronomical characters, but now you have incorporated the disease resistance to it. But it will take 8 to 10 years to develop, and again 3 to 4 years for its release. So ultimately, with that 12 to 13 years, the world population will again change. Again, new disease will come, because the target that was taken by the breeder, it was actually 13 years back. So everything has changed. Technology has also been changed. So again, when actually the product will come in my hand, again I am lagging behind. Again, I will be able to find some lacuna in that variety. Okay, you can take mutation breeding. Again, it will take from mutation experiments. You have to make the mutant generation, then you have to select them, and following each and every process. You can see this pictorial diagram here, very good representation. It will again take 8 to 10 years to develop the variety, and then again 3 to 4 years for releasing it. So ultimately, whenever it will come to the farmers, actually many things has already happened to the world. Suppose if you go for transgenic breeding, sometimes people have some misconception that, okay, transgenic breeding or maybe QTL mapping and then QTL transfer, everything, marker assisted breeding, they are very quick process. But I want to give my note here. That means these are some misconceptions, because for standardizing all those protocols and standardizing that transgenics and its field trial, everything, again it will take 8 to 12 years. And for suppose marker assisted breeding, in most of the books it is written that in marker assisted breeding it will take hardly 3 to 4 years to make a variety. But another thing, if you are going for a marker assisted breeding, actually the information that you have to generate prior to marker assisted breeding, that is the association of your trait of interest with the marker. But to generate that piece of information, that means your trait of interest is related to which marker, it will demand that you have to make one mapping population. Then in that mapping population you have to go for statistical, that means you have to go for genotyping, phenotyping, statistical analysis, everything. That means just to generate that piece of information, you need to you have to deploy almost 4 to 5 years there. So now what is the resultant? 4 to 5 years to develop the information, and then again 3 to 4 years for developing the variety. So actually these are same. Okay, actually with that 10 to 12 years, world population will again change, world environment will again change, everything will change. Okay, so nowadays a very new concept, genome editing, very popular, CRISPR-Cas. Okay, so I can share one of my experiences about CRISPR-Cas. Okay, genome editing or CRISPR-Cas, actually you will be edit the genome, most probably the better way you will be able to you will be able to delete some gene, or you will be able to stop the expression of a gene. But CRISPR-Cas has come to the market just 10 years back. Okay, but and that very famous experiments of tomato that using CRISPR-Cas, we all know about that. But I have one question there. Suppose those who are field workers, those who are doing their the plant breeders, those who work in the field condition, actually plant breeders' main goal is the yield. And what is the nature of yield? The genetic nature, it is a complex trait, and many times plant breeder actually deals with the quantitative traits, that means who are polygenic in nature. So if you use this genome editing, how many genes will you be able to edit at a time? Because those who are practical workers in the field, not in the laboratories, their importance is that I have to deal with a number of genes at a time, those traits who are polygenic in control. But I have a doubt whether this kind of technologies will be able to handle all these polygenic traits nowadays. So it will take time to. I'm not against this, but I think lots and lots of research is required to deal or to handle these polygenic traits by these genome editing techniques. Okay, at a time, suppose 50 genes, at a time 100 genes, like this. So as this technology is very new, is very young, just 10 years ago, the debut of CRISPR-Cas we have seen, we have observed. But this is a question, though, for the practical plant breeders. So okay, now those who work in the field, for them a very good example, people has devised a new technology that is for speed breeding, that I was talking about. That means I have to reduce my time. I cannot wait for 12 years, 13 years, 15 years, but with no or minimum mistake. Again, I am repeating my words, with no or minimum mistake, speed breeding. You can see that now people has made that just using some specialized LED lights. Okay, just people has controlled the photoperiodism of the plant itself. And it is it has been seen that you can grow at least 4 to 5 generations of a single crop in a single year. So that is very interesting. No genome editing, no question of biotechnological tools, no question of bioinformatics, no question of highly sophisticated laboratories, no question of involving so much of money. Just you play with the photoperiodism nature of the plant, and you just give the specialized light. You just arrange some lights that the plant can have, and it will grow 5 times in a year. Okay, and it has been in standardization process. Okay, so this can change the world. This technology, that speed breeding, I have shown you. Okay, and this speed breeding, this approach use the controlled environmental condition and prolonged photoperiods to produce up to 6 generations. I was telling 5 generations, but actually theoretically you can get the 6 generations in a particular year. Okay, as opposed by the traditional 2 or 3 times growing in the natural condition. So this is speed breeding, and people are trying to adapt this. Okay, so that means, suppose why do people get results while working in Drosophila so quickly or in microbes? Because their multiplication rate is very, very high with a very short period of time. But in case of plants, I have to wait for a complete year to grow just to see my segregants. Okay, so people has reduced the time. But now what is left, that is, how can I get everything with no error? How can I minimize the error? And I have already told you, okay, the CRISPR is also has come to the market. People are trying, okay, whether, but still there is a question. That is my own opinion, that whether it will be able to handle 50, 100 or 200 genes at a time or not. Very good option nowadays again coming back, that is the mutation breeding. That means application of nuclear energy in the field of agriculture or in the field of plant breeding. Okay, and marker assisted backcrossing, I have already told you. That means, okay, it can reduce the time with minimum error, but the problem is that you have to develop the piece of information. That means the linkage between marker and trait of interest in a particular population, and you have to develop that mapping population. Okay, and it will also kill many time. And again, another problem is there also in marker assisted breeding. That means, suppose you have developed the information that is marker X is related to my trait of interest, like suppose nitrogen assimilation. Marker X, suppose it is an SSR, it is related to my trait of interest, nitrogen assimilation. And you have developed that piece of information in a particular mapping population developed by you. So somehow, if you are working on a different population or a different ecotypes of that particular crops, these piece of information may not be valid on that. Okay, so again, for your target integration procedure, you have to again develop the marker and trait association information in your own material. Okay, not just blindly. Suppose someone has told that, okay, X marker is related to this trait. Blindly, it is not applicable universally to all the varieties belonging to that species. No, we have to get it again. So that's why people has again trying. Okay, all these technologies already been practiced for a long time, at least 10 or 20 years if I consider the latest. Now this is the era of AI. That means how artificial intelligence can help you in this regard. Okay, that means how can I minimize the error? Actually, what is the actual sense here? That means I want to win over the environment. You think very critically. You just close your eyes and think very, very critically. What is the actual motto? Actually, I am trying to win over my environment. I don't want to be bound by the environment. Being a plant breeder, being a plant scientist, and with no error. Okay. So how can I do that? How can I win over that environment, and how I can achieve that with no error? So now then the question of bioinformatics comes. Bioinformatics, I will not talk about like that. Actually databases. That is very important. Then bioinformatics comes. Whatever the information that has been published, that has been generated by the world-class scientists in very, very reputed journals, we need to assemble, we need to store all those information very systematically in a single database. That means whenever I start my journey for a new plant breeding program, I can just in a single click, I can have the idea that what has happened just before me, each and everything in a single window. And it is possible whenever you are maintaining a database. Sometimes people think that, okay, bioinformatics is only doing something with only DNA or protein sequences or RNA sequences. Not like that. Actually, all the information in a single window information of all the publications, all the scientific achievements in a single place. Okay, so that is also a Herculean task. That's why there are many agencies, they are dedicatedly doing this. Okay, and sequencing and the annotation of the genome sequence, this is a part. Okay, and that's why we are able to get all this information at a time. And I have just shown you this one of the examples. That means suppose in rice plant, and at which chromosome it is present and how it is getting expressed. I will show some live sessions here. Okay, by working with some tools, then it will be more easier to for you to understand that. Okay, that means, and there are dedicated databases for different crops also. So before starting my experiment, because I may have many misconceptions about the gene, about the trait, I will be able to get everything clear in my mind. Then I will be able to focus on the trait, and then only the error will be minimum. Okay, so there are this international nucleotide sequence database collaboration. Okay, and nowadays everyone knows all these things, this Ncbi, then EMBL, DDBJ. Okay, this actually they are the databases where everything is stored, and they are also interlinked with each other. That means data are shared by them. Suppose one piece of example is present in EMBL, you can access them through Ncbi also or through DDBJ also, like this. So, and there are some specialized databases also for individual plants, or maybe a group of plants, or maybe a family of plants, like this. So I have shown here, you can see, that means for many organisms, and under that organism, under genus, under species, under varieties, under ecotypes, you will get the information. And the database is not complete right today. It is being growing up. Actually, database, the scientists are doing all the are working throughout the world, and they are depositing whatever their discoveries, and they are being integrated in that particular database every day. So in a nutshell, you can call it as a data cycle. Now it is nowadays, this is the era of open resource. If you want to publish an article, you will find most of the journals, They are just going for open access. Okay, that means the motto is that knowledge is for all. Previously, it was that okay, for some journals you cannot see the information without taking the subscription or like that. But now the scenario is changing. That means knowledge is for all. It is open access, and each and every information is available, should be available to everyone. That's why you can just prepare your brain, you can prepare your mind before doing your experiment. So what is that data cycle? That means you have to generate the information. Everywhere these kind of nucleotide or protein sequences are shown, it is not only dealing with protein or DNA sequences. Actually, all data, including the phenotypes, everything you can generate, you can submit. And there are specific agencies like Ncbi, DDBJ, EMBL. Okay, they are making the archive of that. They are classifying. There are curators. They are classifying all those data, and they are sharing throughout the world. Okay, and from that data, and at any point of time, you can retrieve the data and you can analyze the data, or also they are also analyzing the data, and they are developing some databases. And these databases can be reused as secondary data by any researchers, and you are welcome to publish all those analyses. Okay, that those in silico analysis, and this is the data cycle. So using that data, you have suppose again made some discovery, and you are again submitting that paper, and somewhere if in a very reputed good journals, again that will be added or that will be integrated as a new data or new information to the database. So that means from a single data, every time it is being cycled, it is being reanalyzed, and it is being deposited, and from a single data, millions of information are being generated. And this is the beauty of bioinformatics. This is the beauty of this data cycling. So what actually this life science machine, what are they doing? Suppose I have taken the DNA sequence of a whole genome sequencing of rice or wheat. It has already been done. Okay, and it is for public access. Anyone can access that. But by seeing that raw sequence, I cannot conclude anything. So that particular sequence has to be converted into knowledge. Okay, only when that DNA sequence is converted into knowledge, then only I will be able to decide, and that decision will help me to minimize the error. Okay, so that means that DNA sequence should go through many scientists, many people, through analyzing through many softwares, and after validation from the other databases, and it will go, if required any curation, people will curate that, no problem. And a number of processes, everything. That means after suppose that DNA sequence, you have taken this as secondary data, and you have done another research, and you have published something, then it has turned into a knowledge. So data alone cannot give you anything. Actually, it has to be analyzed by other researchers, and then it has to be transferred into knowledge. Okay, and I have just taken the example of EMBL databases, because most of the seminars that I have attended, people only many times they show Ncbi. That's why I have decided that okay, I will show EMBL database. Okay, that is European Molecular Biology Laboratory. So the information that you can get from that European EMBL EBI is like that. Okay, it will be much more, actually, from human to plant species, everything. And you can have the DNA sequence. You can, from that DNA sequence, you can get the protein sequence. From the protein, you can get the probable protein 3D structure, or whether that protein is having any interaction with some chemical or not, or that protein-protein interaction. Everything you can do on that web resource itself. Okay, and that is very simple as running in any social network. Okay, it is very simple. They are so user friendly, and you can see the global impact here. Okay, and I have just taken this screenshot from the real-time visualization. That means throughout the world, how many people at that particular time. I have taken this photo on yesterday, sorry, no, day before yesterday at 11:40 a.m. and see throughout the world, people are doing research on these data resources, just on EMBL data resource. You can see the real-time data. Okay, how many people are working at that particular point of time? Okay, this I have just shared this. So people are trying to convert this data into knowledge. Okay, see, that is very common format, and this is very important. Suppose whenever a bioinformatics database has been maintained, how can I use that? That means you can go many ways. In a search option, suppose you can select the organism first. Okay, then you can see, suppose it has written mus musculus, that means house mouse. Okay, so I have written this particular use or specific scientific names. So from that particular database or data resource, suppose you can see that where the mouse is. Okay, so from this, actually through you will be able to retrieve your data. Actually all are interconnected. Okay, suppose why are all the species are interconnected? Because suppose you cannot select the organism, you can just write the name of a gene particular, and which is suppose multi-drug and toxic compound extrusion gene, then it is not a species specific information. Suppose this is a very common gene which is present across the species, so then the results will come not only from a single species, from all the species at a time. That means based on your query, okay, based on your query you will get the result. Okay, and suppose you can type the name of an organism here, name of a gene here, name of a protein here, anything. So it will be retrieved from the data house, and by this way you will be able to, by the graphic view, you will be able to visualize that particular gene also. Or maybe if it is a complete species, you see the complete set of chromosome that has also been shown here, and if you click anywhere of that, you will be that particular genomic portion will be zoomed, and you will be able to view that. Okay, so you can browse the genome, you can blast the genome, and from that, whatever the publication has been made, you can have that in a single window. Okay, but this is the data resource. Okay, I was just retrieving the data, but now I will enter. That means how can I analyze the data? Okay, so there is Ncbi Genome Workbench. You can download it, no problem. You can download it. It's free, and in Ncbi Genome Workbench, you just take the particular data of your interest. Suppose maybe gene sequence or RNA sequence or protein sequence, and you can do. You see at the right side of this. That means all these tools you can apply in that Ncbi Genome Workbench. That means one portion is you are taking the data, and it is secondary data because someone else have developed that particular data, and you are just using it. So this Genome Workbench, you take your data in the Genome Workbench, and in a single window you will get all the tools, and you will be able to do your research. Okay, another important thing, suppose just see at the lower portion, in many publications you will find this kind of picturization. Suppose they are handling a single gene, they are working with a single gene, and this kind of expression view, that means in which organ which particular gene is expressing and at what level. Or maybe suppose at this is the chromosomal viewer, that means your gene of interest, it is present in which chromosome. How can I show this? Okay, suppose you are working with 44 genes at a time or 250, and you have to show that in a single karyotypic map. So how to do that? All these things you can do here. That means this web-based resource. This is the bioanalytic resource of plant biology, bar U Toronto. It is developed by University of Toronto. Very useful bioinformatics resource. I will show this later on, and you can just note down the web address of this. Okay, and from here you can generate this kind of pictures for your thesis or for your research or for your presentation. Okay, this is one of my students' work. I can show you, and I will show you that how to do all these things. That means how to do these interaction thing, how to take the gene interaction, how to get the gene interaction, how to get this gene into protein interaction or gene into any chemical interaction. Okay, this will help you to design your experiment, design your plant breeding experiment very smoothly. So here we have done, we are trying to develop a functional linkage. Functional linkage means that is gene into gene interaction or protein into protein. That means functionally they are interacting. They are functionally linked. That's why I have used the term functional linkage, not that genetic linkage. That means where the genes are present side by side and they are co-segregating. Not like this. This is functional linkage. That means they love to interact together. How to do this? Everything I will show today. And I will just show actually this paper that how did I do this research. That means from secondary data source, from bioinformatics resources, how can I sort out a gene of my interest and I can go for marker-assisted breeding. Okay, and now we are living in this world which I am showing in this slide. This is the era of IoT. Okay, that means Internet of Things. Things may change. That means Internet of maybe agriculture, Internet of medical things, Internet of biology, Internet of anything of you want, because actually the storage, everything will be in the internet, and anyone can access that. Okay, at any point of time. So if each and every data resources are connected and they are stored and everything is present in the internet, just by devising a simple machine or making a simple app, your plant breeding selection, plant breeding mainly the selection of superior progenies or selection of superior genotypes will be very, very easier. Okay, suppose you are having some desired characters based on which you are going to select your plant. You are having a segregating generation. Okay, so you can take the help of genotyping, no problem for selecting those. But for that, you have to take the DNA, you have to make the primers, you have to then take them to the laboratory, then go for DNA extraction, then you run the PCR, then you visualize the band, then you take a decision whether it is present or not. But not only the genomic databases, there are phenomic databases also, phenotypic databases also. That means if you are having, suppose 1 million picture, okay, for a particular variety for different conditions, okay, suppose at which temperature the color leaf, what is the color leaf, suppose at what kind of physical appearances, what is the actually the biomass, everything, if you can store it, and whatever I am saying, that is IoT, and if your device is connected with that particular IoT, you just take the photograph of your segregating population, and that AI will tell you, okay, suppose out of 5,000 plants, suppose 3 or 4 may have the desired trait of interest. So that means in the field condition, you will be able to take the decision. by using phenomics. Okay, so this is our two latest articles regarding, that means how can we apply this new technology of intelligent robot, okay, for plant protection in ecological. That means if you are having the disease database and it is present in the internet, that means in IoT level, and you have devised a simple just app, because each and everyone we are having the smartphone nowadays. So just you take the picture of that particular plant and you just assess whether it is a diseased or not, and what is the disease and what is the causal organism, everything at a single time. So it will help you to judge whether I will select that particular plant or not. And there is no need of you to go in the field also. The robot is can do the job for you. You have to take the decision only. Robot will sort out, okay, you can take this, you can take this, you can take this, but actually what you have to take, that particular decision to taken by you. Actually artificial intelligence in true sense is not artificial intelligence, it is artificial narrow intelligence. Okay, and in another article that we have also published, that means just by this imaging and by phenomics, we can use the deep learning or machine learning application, that it will help you that whether a particular vegetable or fruits, whether it is diseased or damaged, or it is having the export quality or not. Okay, so in a very short period of time, with no or minimal error, you are able to select the particular individual that you intended to select. Okay, but with the help of AI, just only you have to devise one app. Okay, because again I am telling, whatever the technology comes, no technology could replace the traditional plant breeding method still today. That completely traditional plant breeding methods have been resolved. No. So these works, that is suppose diallele, line into tester, they are still relevant for judging your parent, for judging the best genotype, for judging the best combiners, everything. They are still relevant. The tools that are going to use, maybe starting from your genotyping to AI, everything that are just sharpening your tools. They are not replacing your tools, they're just sharpening your tools. Okay, and as I was talking about, that means AI based phenotypic trait estimation, we have already devised one patent over that. That means how can you select the particular genotype? Actually, you make one mobile app and you connect that mobile app based on a database. Okay, and that in that database there are millions of pictures. Okay, and you can enrich the database by submitting more and more pictures, or you can create your own database also, and from that you can estimate the phenotypic traits just by taking the picture. That means another advantage you are taking here, that means there is no need to destroy the sample. You should not cut any portion of that particular plant. Okay, you should not take some portion of root or leaf or anything. No need. You just take the photograph, and you are having the phenomics database, and your device is connected with that phenomics database. You will be able to select the plant, because the ultimate goal of plant breeding is just selecting the plant, selecting the best one. And if I can connect that particular device, that particular app, along with some genotypic bioinformatics tools also, the thing will be more easier. That means you will be able to judge whether you are doing the right thing or wrong. I will show you. Okay, so there are many bioinformatics tools, and it is not that each and everything are getting developed and they are being updated every day. So it was just phenotyping part I have shown you. And in the right side of this slide, just you can see, that means another patent that we have taken. That means by using AI, you can just design a, that means from a say from a very big set of nucleotides, you can just at a very small, very less time, you are able to design, or you are able to select a set of polynucleotide sequence, just like designing a primer in a more and more error free way. Okay, and we have also patented that. Okay, though you are there are many tools are present here. So now just here I will just stop sharing the screen. Now I will show something live. Okay, then again I will come back here. I don't know what is happening. Okay, so are you able to see my screen?
Yes.
Okay, look, I am typing bar U Toronto. I will just show you. Okay, so if you are able to visit this website, you can do your own also, no problem. There are several tools. There are several tools. It is not possible to possible for me to show each and everything just in one day. Okay, there are many tools. I will just show you one new concept, that is ePlant. So whenever in the real world, when I look at the plant, being a plant breeder, there may be many questions may arise in my mind. Okay, suppose okay, what at which condition, at what temperature, what is the expression of a particular gene of this plant, what may be actually what is going on here. Or maybe suppose if I take out that plant from India to maybe suppose in Saudi Arabia or maybe to USA, what will happen to the gene expression? Okay, so many things we try to anticipate, but we don't have the answer. So I am just showing you the ePlant version. Okay, that means here you can get each and every information, and how to retrieve that I am showing you. Okay, ePlant is for maize, poplar, tomato, camellia, soybean, potato, barley, medicago, eucalyptus, rice. Okay, there are so many plants available here. Just for example, I am just taking the view of, okay, ePlant rice. Okay, as the rice is a model plant, I am showing you the rice. Okay, so here these are the tools. These are the tools that you can visualize by this ePlant. ePlant means this is an electronic plant. Okay, this is the virtual plant. Just by clicking at different parts of the plants, you will get from genotype to phenotype each and every information in this ePlant, which is not possible in your real plant. Okay, then you have to collect, you have to take the DNA or RNA, then you have to go for laboratory experiments. Suppose there are many features, maybe word E expression. Okay, there are many things I'm just showing you, but it must be visualized for a particular gene only. Suppose for example, I have taken one gene. It will be downloaded to the system. Okay, suppose this gene of rice, this is OS 01G 01080. Okay, from that means this is the gene from chromosome 1. This is the genomic DNA portion. Okay, so I want to know. Okay, so that I was talking about. Okay, so for that particular gene, I want to know the expression. Okay, you can see that this very particular gene, its expression is mostly in the, suppose, are you able to visualize or shall I zoom? Yes, you see the red means it has been more expressed. So what is the tendency of that gene? It is being expressed in the seedling root. It is completely red color. So already you have got in your mind, if you that means your brain is not blank in this case. That means you have got the idea. That means this gene is having the tendency to be expressed in the seedling root, and where in the inflorescence also, but they are being less expressed these stages of the inflorescence. Okay, and suppose in seed there are different kinds of seed stage of seed development. Okay, that means in mature leaf you can see that different stages of seed also. What is the level of difference in the expression of this gene? Okay, so you are already having in your mind the idea. Okay, suppose that means that gene, if I want to improve some root character or these things, so I have to, then I can target this gene as it is being expressed in the seedling root or in the inflorescence or in the mature leaf. So it is maybe related some works, some functions, okay, in that organs, or otherwise you can click here, tissue and experiments. Okay, okay, particularly you can see the tissue here. That means in which portion. See, in the coleoptile especially, this gene is expressed more. Okay, and when the coleoptile matures more, the expression is reduced. Suppose I want to know the genomic chromosomal information. That means the gene that I have taken, it is present here. This is the chromosomal location in chromosome number 1. Just it is present in the here, present here. Okay, or maybe if you want to view the interaction, yes, that means this particular gene, it has been shown here in the middle, and it is having interaction. This is the epistasis, so interacting with these genes. So I can I may query that what is this gene. Okay, so everything. Okay, so you know the name of this gene and protein interaction coefficient. So if you just copy the gene name and you paste in the rice genome browser, you will be able to know. Okay, to that means the epistatic interaction of your gene of interest to which other genes. Okay, you can see. Okay, the sequence viewer also. Okay, that means. Okay, the particular sequence, if I click here, I will be able to download everything. Okay, but as I am running out of time, I need to show something more. Okay, another actually the feature that world EFP I want to show you, that is not till today not available for rice, but it is available for Arabidopsis. That will be very interesting for you. Yes, suppose any gene of example, I have taken one gene and have loaded the gene. It is being loaded. You have to give some time to load a particular gene there. Okay. So Arabidopsis, for Arabidopsis, all the features are available, but for other crops, the features are being updated. They are under process. Okay. That I want to show you. Yes. Look, the gene that is ATG G602200, that particular gene I have selected. See the expression of this gene, okay, in different locations of the world. You can see the same gene, suppose in Poland, suppose in Germany. Okay, that means based on the different climatic conditions, suppose in Finland, you can see United Kingdoms, it is the redness is more. That means this gene of Arabidopsis, look here, the expression is more than this place. Okay, so as much as possible, as much as data that will be deposited here, that means the total world map, world expression map for that particular gene will be completed. So again, you will have the idea. That means that gene is whether at which weather condition this gene, what is the pattern of gene expression of that particular gene. Okay, so that means it will help you to take the decision. Okay, again I can show you. Okay, the particular gene, you can see the whole organ view. That means where the expression is much more and where the expression is completely absent. Okay, you can see from here. Okay, you can see the chromosome viewer. That means at which location of the chromosome the gene is present. Okay, or you can see the interaction of that gene with other genes. That means the epistasis at molecular level. Okay, you can view the particular protein sequence, that 3D model of that particular gene product. Okay, so those who are interested in protein engineering, it is very important to them. Okay, and from there you can suppose you can, yes, molecular surface, everything. Those who are interested, they can have the information. Another important, yes, that particular gene, as we have seen, that means their organ specific expression. See, now it is the cellular localization, cellular location specific expression. That means this particular gene is expressed throughout the world, but not suppose in mitochondria. Okay, here in Golgi, suppose in endoplasmic reticulum, score is 0. That means that gene is not expressed in. That means it is having no role in endoplasmic reticulum, but mostly expressed inside the nucleus. See, the score is 66. Okay, so how can you get the idea of a particular gene? This is the beauty of bioinformatics. All the information at a time. Okay, so okay, you can get the gene information at a single place. Yes, you can see the gene, the DNA sequence, the protein sequence, everything in the same place. You can see the pathways, as there is no pathway still today reported, so it is showing that okay, no pathway is found. Okay, you can see the heat map viewer also, and wherever you click, you will get. That means again, actually at which tissue and where it is being expressed. So suppose you are getting this gene. For example, I am telling. Suppose you have got this gene of interest that is being expressed in that is related to suppose yield, or maybe whatever you take, suppose seed number, maybe for that. So you can take out this gene, and now previously you have already known. That means which are the tissues where I can get the maximum expression, and that's why by that way I can target that particular gene, or that as the genome information is also present, I can design the primers. Okay, that means yeah. And if I go for real-time PCR, and as I already know, suppose it is the maximum expression level is suppose in root. Okay, you take the RNA from root only, and then you will get the maximum expression there. Okay, so previously you know everything, and it will help you for a better judgment. Okay, so okay, already 1 hour is over. Let me share my screen again. Okay, so if this is there is a demand of the house, then I can take some other lectures also. That can also be arranged. That means they are by specific using of specific tools for a single lecture. That can be because in a 1 hour class, I think it is not possible for me to show you all the tools. I mean, that means how to design primers, how to run the blast, how to take the decision from blast. Okay, how to know the open reading frame. Everything I can show you. But at the last, my personal opinion. That means whatever we are doing in the lab, it must reach to the actually to the land. Okay, whatever technology we adapt, okay, it should not be restricted to the laboratories only or in very good papers only. It must reach to the farmers. Okay, the technologies, the products of the technology must come to the farmers. So actually, what I can speculate that much more very much sophisticated tools, techniques that has been designed, but their products are hardly seen in the market. Okay, so I think all the plant breeders should focus on that. Okay, and bioinformatic tools will obviously help the plant breeders to sharpen their tools for selection. It will never be able to, for my opinion, to able to replace all the traditional tools. It is not possible actually. Actually, it will sharpen the tool. So the message from me, that is the let the farmers be happy to keep the planet smiling. Okay, so do whatever you want to do. I think now it is the time to the back to the phenotyping again, because all the genotyping everything. So that's why today's AI based technology also you will find. That means they are just integrating everything from phenotype to genotype, everything in a single device, in a single app. That means in the field itself you will be able to select your plant. That I have already told you. Okay, so this is the only message I want to give to the to my fellow plant breeders. That means whatever we do, whatever we do our research, that must be with the focus to make our farmers happy. If the farmers are happy, then only our planet will smile. So thank you all. So today's lecture is over. So thank you all. Namaskar.
And happy Rath Yatra.
Okay, so I'm requesting the host, ma'am, to conclude over the seminar or the webinar.
Thank you so much for this very nice session, sir. If anyone has any question, you can ask.
Okay, ma'am. So I think if there is no question, then we can conclude here. Okay.
Sir. I think no one have any question, sir. Thank you so much, all the participants, for your active participation. Let me inform to all that this complete session will be available on our Research Circle YouTube channel. So if you want to watch it again or want to share it to somebody, please do visit the Research Circle YouTube channel. Thanks. And last but not the least, thank you, Dr. Sandeep Devnathan, for taking out time for session. With your permission, I would like to end today's session here, sir.
Okay. Thank you, ma'am. Thank you, everyone.